S2 ya, dan ada S3 juga. Silakan bisa menempati yang di depan-depan saja di sebelah sini. Ada di belakang gelap. Biar kita bisa segera mulai. Karena sudah lagi cari malu kegiatan kita mulai yang di bagian belakang mungkin bisa maju boleh. Yang di sebelah sana 3 masih kosong, 3 masih kosong. Silakan Kak, kita maju aja. Pasti. Ya, kebidanan ada, kemudian MIB ada, kemudian IKT ada, mahasiswa S3 ada lagi, magister mana lagi yang ada ya, sama ada yang di lain. Silakan.
Good afternoon, everyone. It's my pleasure to be. I'd like to thank everyone who's introduced me and the subject. They're very generous introductions. As I think was said, I'm here visiting Indonesia because we have a joint project. Also at Irwin, there's this worm seed project with worm drug resistance, soil transplants. Southeast Asia, a joint project between UK, Indonesia, Philippines, and Malaysia. And some aspects of that work come out of previous work I've done. My particular interest is Strongyloidiasis. And that's what I'm going to speak to you about today, particularly how to use genome sequencing of parasites in a non-destructive fashion to try to understand the biology of parasites. Is this working? You hear me at the back of the head. Okay. I also have a tendency to speak quite fast. So if I'm speaking too fast, and you can't understand me, just put up your hand, tell me, and I'll slow down and get past. So what I'd like to talk about today is this work. And this is mainly the work of a PhD student of mine in Liverpool. Yu Chen Lu, who's a Chinese student, came to Liverpool now back in time. We worked with a whole series of colleagues in Cambodia, Bangladesh, Thailand, on this work. Is that okay? Is that better? Now it's better, okay. Is that the best format? That's a bit square. Is that okay? Actually, shouldn't be. It's fine. It's fine. So how do you push this? But technology is against us today. But actually, I'll point with that. Okay. So Elsa gave a very nice introduction to Strongyloides, and I thought I'd show you the parasitic worm. This is from the species of rat. So this worm lives in the small intestine. It's about 2.5 millimeters long. This is its head here. It's a great big long outfit. This is a filariform esophagus. So it has no bulb in it. That goes right here. Then here is the vulva where the eggs come out. There are a couple of eggs in this worm. Then it goes down to its tail. And really, most species of Strongyloides are pretty similar. The morphology is very conservative, very similar among the species, which really is why we've moved to molecular approaches. Professor Elsa also described the life cycle, but I would like to just talk about that a little bit more, because thinking about using genetics to understand what the parasite is doing, you need to be aware, one needs to be aware of the genetics of the life cycle. So in Strongyloides, the parasitic stage is a female-only worm. So there are no males. And this female worm reproduces by parthenogenesis. Do you know what parthenogenesis is? What it means? Somebody tell me what parthenogenesis means. It's a too scary question. Has anybody heard the word before? Parthenogenesis. Do you know what it means? Anybody? Okay. It means virgin birth instead of translation from Greek. But it means that this female will reproduce by mitosis. So it makes its eggs, not because there are sperm from male, it just makes eggs by mitosis. So her offspring are genetic copies of herself, and all of her offspring are identical. Okay? So these worms come out of the host. They're male and female, and they can do one of two things. The eggs that come out of the host of the feces, they can develop directly into these infective larvae, third stage larvae, similar to hookworm. These persist in the soil. When that soil comes in contact with the skin, they'll penetrate the skin, they'll migrate through the body, getting back to the parasitic stage in the gut. Now in the Strongyloides, and that's a little bit like hookworm, actually, except hookworm is males and females. Now, Strongyloides is rather unusual because it also has this alternative option, which is where the male and female eggs here. What's that? Somebody needs to stop that. They're messing up my slides, aren't they? Okay, so these eggs here, they can grow into male and female free living nematodes. These are adult nematodes that are in the feces and the soil. These males and females mate by sexual reproduction, just as we do, and they have offspring develop into infective larvae. So this life cycle is entirely asexual, there's no sexual reproduction. But if the worms develop by this route, then there's sexual reproduction. But this sexual reproduction is facultative. That means it's optional. It doesn't always happen. So I thought I would talk, and I'll talk slightly quickly because we've spent time not having any interesting, about two different studies. I'll speak a little bit about Strongyloides rati in rats in the UK as a population genetics we have done with that to understand how those parasites are distributed in the host population. And then I'll spend most of the time talking about Strongyloides stercoralis in people and dogs in Asia. So in the study where we looked at Strongyloides rati rats in the UK, about 75% of rats in the UK, Rattus molecules, are infected with Strongyloides, a normal parasite. But we collected parasites non-destructively. And what that means is we didn't actually catch and kill any rats. All we did was we pick up rat feces in places rats were. Those feces have eggs in them, Strongyloides eggs. We cultured those in the lab. Infected larvae grew. We took those individual worms and then we whole genome sequenced those larvae. Okay. So this is non-destructive parasitology. And because this has a path to genetic reproduction, genotype of the worms in the feces is the same as the genotype of the mother. Sample exactly what's in those. But this parasite, people who are on the course last week, what this means is a high quality reference assembly. So there was a assembled genome. Then we sequenced, DNA sequenced these larvae, compare them back to the reference assembly, and look for variation, so single type polymorphisms, so SNPs, to measure the variation among these parasites. At the same time, we genotyped the rats. So because there's some cells from the rats in the feces, we use some other genetic analysis to identify the rats. So where there were two big components from the same rat, detect that. This is where we did the sampling. So this is the UK, Europe is down here, it goes up here. And we're in the southwest corner of the UK. We sampled at 3 places near the city of Bristol, a farm here, a waste treatment waterworks here, and a rubbish dump. So this is separated by the map. So we have 90 parasites from those 3 sites. I'll just summarize what we found out about these parasites. So first of all, the physical distance between these places. So these 2 places are one side of the river, very close together. The one on the other side of the river is much further. If we look at the genetics, and this is a measure of genetic similarity of the rats, what you see is the rats are about a similar amount of genetic distance between the rats' two sides. So it's linked. This line shows how different the rats are. So they're about the same difference at each size, sort of equivalent to the geographical distance. And that's probably what you'd expect. As animals are further apart, they mate, nest together, so they'll get genetically different. But the parasites are quite different. So this is the FST, which I'll explain later on, but it's a measure of genetic separation between the parasites. The parasites between the 2 sites on one side of the river had no separation between them. They were a single population, which might be partly to do with how their life cycle works, but it also might be due to the effective migration of the parasites. But the parasites on the other side of the river really were a little bit different as well. So that's a summary measure. I think this will show those 90 parasites altogether. So this is a neighbor joining tree. So we've taken the genome sequence, all the parasites. We've looked for single nucleotide polymorphisms, so sites, bits of DNA that are different among the parasites. And we've used that to calculate the genetic similarity between every parasite. There are 90 parasites here, and then we built a tree which shows the similarity between every parasite. So every tip here is a different parasite. And there are 90 of them. What we find is they separate really into these pattern 5 clades here. It's great big clades into which many parasites from the 3 sites belong. And we have these other smaller clades. And really the reason we're getting the genetic similarity amongst these parasites at different sites is that genotypes in this big clade are present in all sites. That might be that it's a spread of an asexually developing genotype across Southwest England. We've also measured admixture, which again, I'll explain a little bit more in a minute. But this is a measure of the percentage ancestry of the different parasites. So each column is one different worm. And this computer program calculates the likely ancestry of those parasites. And it shows it by color. So all these ones in blue, say they are ancestors from a single group, the blue group. This one here has ancestry where it's about 30% red, but 70% here. So these are ones where there's 2 colors in the column. It's a mixed ancestry. And what you can see here, without going into the details, is lots of the parasites have a single ancestry, a single color. It's a minority of the parasites that have these mixed ancestries. And what I think the reason this happens, I think, is that in Strongyloides rati of the UK, there aren't sexual stages. So you remember in the life cycle, I said there was facultative sexual reproduction. The parasites in the UK seem not to have that sexual reproduction, so they're always consenting. We have high heterozygosity, so it's any particular locus, any gene. We might commonly have 2 different alleles, so this is different homozygous. That's again because it's an asexual population. It's never any sexual reproduction. We also have extension linkage disequilibrium, so that is linkage between markers because there's no sexual reproduction, no meiosis breaking that up. We have little admixture. So these asexual lineages, so these parasites that Strongyloides rati exist as lots of different genotypes of Strongyloides spread over the rat population, but they aren't mixing by sexual reproduction because they're asexual. This has some consequences with population genetic structure. Now, that's probably quite a lot of genetics, quickly, but I just say that really just to give some context to the work about talk about now, which is Strongyloides stercoralis, people and dogs in Asia. Really, here we've used the methods we developed, the Strongyloides rati, to ask a question about parasite transmission in this other species. And you saw this actually on the slide Elsa showed of Strongyloides stercoralis from the CNC, because that had a human involved in the life cycle, but it also had a dog on the life cycle as well. And that slide is reflecting a long-standing question in the literature about whether Strongyloides stercoralis is a parasite shared between people and dogs. So that by which means people could infect dogs, but dogs could infect people. Or whether in fact there's 2 different parasites there, one that infects people only, so people infect other people. There's genetically different parasites where dogs only infect dogs. This has been unclear in the literature for a very long time. But really, the problem is that parasite looks the same. You look at it, it looks the same, but the morphology is very hard. It's very insensitive way to tell parasites apart. So that method of analysis really wasn't very helpful. So really, this is the question that we set out to address. So to use population genetics to ask whether there were two different transmission cycles. So one for people only, one for dogs only, or whether there's a transmission cycle where people and dogs are infected with the same parasite. Now, this is of academic interest. So I'm interested. I know there's interest in doing this. But it also really matters if you want to control parasites. If people are infected with Strongyloides and you want to cure that infection, cure them, but to stop them getting reinfected, you need to know where that infection is coming. And if that infection is coming from other people, that's okay. So you're treating people. But if the infection is coming from dogs, you need to treat the infection in dogs as well. So if you want to stop transmission, you need to stop the source of transmission, the source of infection. So you need to know where the dogs are a source of infection. So it has a practical application as well. Another way of saying what this really is whether whether dogs are a zoonotic source of infection. I'd like to put this question about Strongyloides in context of other human infecting helminths. So Ascaris, which is a large, very common roundworm, again, it's been a long-standing question about whether the parasite in pigs and people is the same or different parasites. So the same problem, the parasites are the same indications. Once you control infection in people, you need to know where the infection is. For quite a long time now, there's been various molecular analyses of parasites, Ascaris in pigs and people. And it seems to show that the parasites are largely host-specific. So one in people, one in pigs. But occasionally they cross-infect and then they introgress. So they cross, but the progeny, the offspring, back across to one of the parents. So sort of integration. You can detect that event molecularly. More recently, there's this question about Drunculus, Guinea worm, which is in West Africa, the parasite of people, where there's been a very quite long-standing and quite effective control campaign. Parasite has always seemed to be people and in dogs. But molecular analysis now show that the parasites in dogs are shared between each other. So that means that people can infect dogs, dogs can infect people. Really, I think what's happened here is that people are being treated very heavily to try and remove the human infection. The parasite might even be evolved to be dogs and so remains. And in both cases, of course, these have implications of control diseases. So just thinking about Strongyloides stercoralis in people and dogs, we're not the first people to put this study. But many studies I find a little bit not convincing. So in many cases, people take feces from people and dogs. They compare DNA. They do a PCR, the ATS sequence, the ribosome RNA. I think this is a bit problematic because it's not completely clear what you're amplifying. You never know whether that sequence you get actually is come from a worm. It might be chimeric, a mix of two pieces of DNA. People also do non-sympatric sampling, allopatric sampling. So they take dogs from one place, so dogs from Malaysia, and they take worms from dogs in Malaysia, worms from people in Indonesia. Those worms are different. What you don't know is whether those worms are different because of the geography, 1,000 miles different, or because they're different hosts. So I think you need to do sympatric sampling, where you take parasites from dogs and people in the same place. So you know the geography is affecting the answer, you know better. There's also a problem, which I won't go into, but people put pieces of DNA in databases, sequences of DNA in databases, and attach a species name to it, but they don't have a very good basis of saying the species name. They usually say the host species, give a parasite species. And this creates a lot of confusion. So I'd always say be quite cautious about the parasite species name when you see DNA database. So that's my portion. The study we did in this part of Asia, so not quite where we are today. So these are the countries that we worked in, so principally Bangladesh, Cambodia, Thailand. And we sampled sympatrically for parasites in people and dogs. And these, this color coding is going to pop up through the rest of the talk, so it's worth getting here now. So this is the prevalence of Strongyloides in people and dogs, so relatively low prevalence systems. Quite a bit higher in people in Thailand, but rarer in dogs, and actually quite a bit higher in Cambodia as well. So parasites from these three countries are ones we've sampled, that we hope whole genome sequenced, just as we did with the parasites from rats. Also in the database, people had already sequenced some parasites from Myanmar and from Japan. So we've added those into our analysis. We didn't generate the data. And we also had some samples from Fiji. And this is slightly strangely, I only found this out recently. There are 5,000 Fijians in the British Army, which is part of the Commonwealth. These soldiers come from, these young men come from Fiji to the UK, and their health is checked when they arrive. Quite a lot of them have Strongyloides, and we had a contact, so we've got the parasites from these Fijians who are in the army. So Strongyloides was found in the UK, but we're treating it as this comes from Fiji, because we're pretty sure the infections from Fiji. Before we get into the results, I just want to give the context of these parasites. Just really what we're asking is, are these parasites, dogs and people, are they host specific? So dogs only affect dogs, or people only affect people, or the parasite shared. This is one of the communities that we've worked in Bangladesh. So this is a slum region of Chittagong, a big city in Bangladesh. Very, very helpful community, but they're very poor. So this is down towards the river. This is wastewater from open drains. There's a lady here with her children there. It's the husband. So she's cooking here. There are dogs here. There are dogs everywhere. Dog feces everywhere. You can see rats. There's goats. Wild boar come out at night. There are chickens. So this area here is absolutely contaminated with feces. All these animals. There is some open human defecation as well, and there is some poor sanitation. So thinking about Strongyloides, it's probably Strongyloides larvae, lots and lots of different species in this area. The children particularly are barefoot. So really what we're asking out for is Strongyloides larvae, do they find the correct host species? Do they infect anything? And this context is broadly similar, I think, in any poor community in the countries we worked in. These are the right conditions for transmission. So direct contact between human skin and nematode larvae species. So people who were on the workshop last week talking about aligning sequence reads. Let me go back to stage. So we had all these worms, then whole genome sequence them, so we have all the sequence data. Then we had to analyze the data. And the first step, people from the WormSeed project will realize is we first of all to align those reads to the reference genome. There's a reference genome for Strongyloides stercoralis. And we actually already got our first hint of what's going on by looking at this. So this is how much of the stercoralis reference genome is covered by the reads from parasites from a dog in Bangladesh, a dog in Thailand, And what you see is that the worms reads from the worm to humans align very well to the reference genome. Sequence reads from dogs actually align less well. So that already begins to suggest parasites from dogs and people are related, just at this initial stage of the alignment. So this is as a workshop, we were aligning the Homunculus reads. You remember it's quite important. This is the reads from the whole genome reads to the nuclear genome, summarized to those that were lots of samples. So one of the first analyses we did was we calculated the FST, which I mentioned. The FST is a measure of the genetic differentiation, genetic separation between subpopulations compared to the total population. Okay. So in our study, the total population is all the parasites we got from people and dogs. And then we've made different subpopulations. And if FST is high, goes from 0 to 1, says there's complete separation between the subpopulations. 0 says there's no separation between those. So we can just look at these median values here across the different parasites. So if we compare human parasites from people with parasites from people, we have a low FST. So that says that all the parasites from people are quite closely related. There isn't any separation. But we can compare parasites between dogs and people, then there's a high FST. So that's suggesting there's a genetic differentiation, genetic difference between parasites from dogs and parasites from people. But if we also compare parasites from dogs with other parasite dogs, all parent parasites, we also see there's a high FST. So that suggests there's different types of parasites. It's a bit hard to understand this. So let's, I think, next slide, let's look at a tree, which I hope will make everything clear. So this is a neighbor tree of the nuclear genome. So we've excluded the mitochondrial genome. So we've measured for each worm, calculated the single nucleotide polymorphisms and SNPs, calculated between every pair, said how similar are those two worms to each other. They've displayed that on this tree. Okay, there's a lot on this tree, so I'll explain it quite slowly. We'll look at this different bits of this tree. So each worm is a tip. So for example, here there's about perhaps 10 or 20 tips there. Each one of those is from a different dog. Each one of those is a worm, a dog, family. So you've got dogs. The color tells you where those worms came from. So these are Bangladesh dogs, Thailand dog, Cambodia dog. So this part of the tree is fairly tight. Okay. So I've expanded it here as well. Clever people, are there any patterns that are immediately obvious to you within this tree? What are some of the conclusions you might think about that tree? Does anybody have any thoughts? Yes, please. Why do you say that? How does the tree show you that? Well, brilliant. You give that student a first class degree. Perhaps you're more senior anyway. Absolutely right. You've got it in what? All the parasites from people are very closely related. Very, very closely related. Yeah. These are all people. We'll come back to this topic. All people. And how long the branches are tell you how close related. They're short branches, which means they're very, very similar. The parasites from dogs have got these great big long branches. They're quite different from each other, but they're also different people. So you're absolutely right. So immediately you'd say it looks a bit like the parasites from people and dogs are different. If the parasites from people and dogs were the same parasite, people and dogs would be all muddled up here, but they're not the same. Absolutely. Yeah, so the parasites from people are very close related. We've got two major branches, the parasites and dogs. But it's a kind of a bit odd, really, actually, because we've got parasites from Bangladesh dogs. Same city, same place, same slum area, but very different parasites in. So the dog parasites are very heterogeneous. This is amazing if you think about it, because these are from Fiji, which is a long way in the Pacific, right through to Myanmar. That's thousands of miles, tens of thousands of miles probably, but the parasites are very close related. That might suggest that parasites are moving quite a lot because so we move a lot in planes and trains and boats. Dogs perhaps move less. And they run around, but they don't cross borders very much, perhaps. So we have this. This is kind of the problem with our colleagues here. So that's a dog parasite, very, very similar to all of the human parasites. Why might that be there? Does our experts have a point? How might we interpret that dog parasite in the red box? Tricky. Well, I think this is a reverse zoonosis. So I think this is a dog that was infected by a parasite in people. Accidental cross-infection, reverse zoonosis. So this one has got a yellow triangle, a yellow diamond shape. We'll see that again. We'll see this again as well. This is the reference genome. This is from a dog in the United States, 1980s. The United States, just about 40 years ago, it's very closely related to parasites from dogs from Cambodia. Interesting. We'll also look at these three dogs with the black dots later on. Yeah. So, yellow diamond, the grey circle, and three black circles. We'll come back to those on another tree in moment. So, let's look at the admixture we looked at the rats. So every column is a different worm. These colors now are the admixture groups the parasites belong to. So they're not the color coding you have before. We've got the color coding up here. So this is Thailand, dogs, that's Japan, Bangladesh, and so on. And again, what you see is lots of parasites from people have no admixture. So it's a single color. Get rather more admixture, sometimes in dog. So the admixture is not consistent. So here, all the parasites from dogs in Bangladesh are from different clades, but there's no mixture with these parasites here, for example, Fiji. I'll come back to this. Just to point out, this is the reference genome which has no admixture. This is the one I think is a reverse zoonosis. And these are the three black dots. We'll come back to it. I'll come back now. So previously, I showed you the nuclear genome. That's the same worms, but the mitochondrial genome. So it's a little bit complicated. It's a tree which is made into a circle. Around the outside, and we've got the same color coding. So people and dogs have a color telling the country. Around the outside, we've got the nuclear clade. So that's clade C, clade A, clade B, D1, and all of this is D2. And it, and then we've got the mitochondrial tree here. Then we've got 8 clades of mitochondrial genes. Okay. So what you see here is that we have all of these clades are parasites from people. If you look at these branch lengths, which are a bit hard to see here, quite short. So again, this is absolutely consistent with the nuclear genome. But lots of the parasites from people are very similar to each other, are very similar to each other. The dog parasites here are on quite long branch lengths. So that says these parasites are quite different types of dog parasites, again, consistent with the nuclear genome. Next. We remember this parasite. Remember this parasite here, yellow diamond. That's the dog parasite. The nuclear genome is the same as the human parasites, or the mitochondrial genome says the same story. So this dog parasite has got a mitochondrial genome, same as mitochondrial genomes of the human parasites. So this confirms to me that it's a reverse zoonosis. The dog is being infected by a person. But these are interesting, these 3 black dots here. You'll remember these were down at the bottom of the nuclear tree. So they had a dog nuclear genome, but they've got a human mitochondrial genome. So they're really agree with other human parasites. This is a little bit similar to the aspirin situation. So what's happened, we think, in the past for these parasites, these parasites between dogs and people, they've mated, they've crossed, and these dog parasites have acquired the mitochondrial genome, human parasites. So they've got their hybrid, so they have a huge dog nuclear genome, like all the other nuclear genomes, they're dog parasites, but they have a mitochondrial genome, human parasites. And so it's an example of introgression. Just so here, this is the reference again. So dog parasites. It's actually rather closely related to other human parasites as judged by its mitochondria. So now we can use this to try estimate some large common ancestors between parasites. So you can really add some time to this. So we calculated basically some assumptions. Obviously, the last common ancestor of the dog and human parasite, I think it's around about this time, so about 5,000 to 12,000 years ago. Interestingly, dogs were domesticated by about 10 to 15,000 years ago. The human parasites, the last common ancestor of the human parasites, was 2,000 to 4,000 years ago, so much more recent. So what we think, our hypothesis, is going on, is that people acquired Strongyloidiasis, dogs, when people began to domesticate dogs. So originally, Strongyloides stercoralis was a parasite. Dogs, perhaps before that, was a parasite of the ancestor of domesticated dogs. We think the parasites were largely asexual, so not very much sexual reproduction, like parasites from rats. When people began to domesticate dogs and live with dogs, there was a zoonotic transfer of parasites into people. And then these parasites have diverged. The parasites in dogs have stayed like they are. The parasites in people transmitted amongst people and have diverged. But there is occasional sexual reproduction between these, and we saw in our work there's a cross infection, so that dog got infected by a human parasite. It's a paper that shows quite convincingly that a person got infected with a dog parasite, but probably less than 1% of infections. Those reproduce, so they can exchange nuclear genomes, they can exchange mitochondria. But after they've sexually reproduced, they go back into their, quotes, normal host, do this impression. We think this is what's going on at the moment. So I started. So what I've said all that, you might say, well, that's fine, Mark, but actually, surely then the Strongyloides stercoralis is two different species. It's one in dogs and one. Well, we've made it phylogeny using the mitochondrial genome, and we find that not to be true. So all of the parasites in the study I've talked about are here on this tree. So they're, despite them having some differences, actually compared to other species, they're very, very similar, essentially identical. They're quite different from parasites in rats, that are Strongyloides filiborni, also mentioned in primates, these parasites in cows and so on. The second species in rats, for instance, species in sheep. So I think it's consistent. This parasite is, in dogs, the same species, but there are two varieties, two types. One that preferentially infects humans, and one that preferentially infects dogs, but not exclusively. So overall, it started with asking the question, is there a parasite in people only, parasite in dog people, parasite in dogs maybe, or parasite shared between the two? I think what we've shown is this doesn't really happen. And essentially, they are species-specific transmissions, but it's not completely separate. There's a little bit of leakage between the two. Some rare events where dogs can infect people, people infect dogs. So that, I thank you very much for your attention. I'd like to thank everyone who's helped us work. All these people have helped me. And very happy to take your questions if you'd like to all. Thank you.
Okay, once again, give a big round of applause for Professor Mark Viney. Thank you, Professor Mark, for the very interesting presentations, and I think we have learned a lot, yeah, about the population genetic of Strongyloides in especially in people, and yeah, any variant, and we found many interesting as well from your presentations, like how the worms can select their way or their methods of reproductions that sexually is actually facultative, and mostly they can produce the asexual part, and maybe it is direct or indirectly will also contribute for the population genetic of these worms. Thankfully, I think human we cannot perform we don't have the options here for the facultative reproductions. All of us are sexually reproductive. Yeah. I cannot imagine if human can do the parthenogenesis. Yeah, 1% of Yuki is hard to handle. Yeah? Can you imagine if we have 10 of Yuki? It's not fun. Thank you very much. Okay, so I want also to say hi for our participants in the Zooms. If you have any questions, please, you can also write your questions in the chat box. And now we have come to the discussions part. Maybe the first sessions, I would like to invite all of you in this room. If you have any questions, both for Prof. Elsa or Prof. Mark, please. Oh, yeah.
Thank you. So thank you, Prof. Elsa and Professor Mark for this interesting presentation. I would like to ask to Mark about the genetic relationship between strongyloides from human and dogs. I actually haven't read the study, but how about the sampling method? The research sampling the dog that live around human or just random feral dog? And is there any implication that previously you showed that basically Strongyloides in human reproduce with asexual cycle, right? So is there any implication on mass drug administration? Thank you.
So the first question was how do we sample? Yeah. So the dogs, dogs that lived in and around people, so they were in the same communities, very much so. Particularly in Bangladesh, if you remember that picture I showed you, it's actually quite different from Indonesia. I was surprised. I don't think I've seen a dog since I've been here. Bangladesh, also a Muslim country, there are dogs everywhere. So they're just wild in the streets. So there are 2, 3 dogs everywhere. So these dogs are living in very close association with people. I think in Thailand people have pet dogs, but still they are in that compound. So yeah, so the sympatric sampling is absolutely relevant. I mean, just think back to what I said, what I should seem to show, these parasites of dogs and people are different. And if there was this situation, dog feces, human feces, this is the right place for that transmission to happen. So this ground has to be covered with dog parasite larvae, but they don't affect people. That's a remarkable piece of biology. Your second question was about mass drug administration, I think. So right? Yeah, in Thailand, I think there is mass drug administration. I don't know to what extent it's used in these communities. I don't think it happens in Bangladesh. And I think even if it does, these communities we're in are, don't think the kids go to school. So I don't think, even if there's a process of mass drug administration, I don't think in this community be reached by those processes.
Does that answer your questions? Okay. Thank you very much, Mark. And then, is there any questions from the audience here? No?
So, it, it.
I don't really know that also, actually. I don't think we really know what the larvae do. And it's sort of but certainly our data say that in Asia, parasites would be interesting to know in Africa because strongly stercoralis dogs now. But it might be quite different then. That whole study needs to be done. It's not necessarily what we're saying here. It's not necessarily the same what we're about. I mean, I suspect the pattern we found here probably carries on through Asia, through Indonesia, as we go from Bangladesh and Myanmar to Fiji. But Africa is quite a long way away, and there might have several evolutionary parasites there. So until somebody does this.
One more question about the stuff. Well, we studied them.
No, I think no. It's a study that needs to be done, particularly in Asia. I think also because yeah, so it's clearly very different from Stercoralis. And you're right. So the literature says it's a zoonosis. It's in primates in Africa. So zoonosis interbreeding in Africa. It's in primates in Asia. There's a parasite people in New Guinea where there are no primates. So I think it's really unclear what's going on. There are quite a lot of case reports of Philoboni blood culture, but I think diagnosis is not very much. I mean, quite honestly, studies do it's the same study as here. So you go across Asia and you look for people, both Philoboni people and primates in Myanmar, see whether it's the same thing. It's a bit more complicated because there's also Stercoralis infection. So people have two parasites, but it needs to be worked out as an I suspect that Strongyloides in Papua New Guinea is probably the version of the law, but there's one paper now that we've seen, but it's not really but there's a whole for the younger generation starting their research careers. Sorry, you as well. There's a whole thing to be figured out in Asia. To what extent Strongyloides zoonosis for primates this might particularly apply with rurally forests, so pretty more exposed to primates perhaps in the cities. I'd love to know the answer, but I'm too old to do the study now.
Okay, thank you very much, Prof. Elsa and also Prof. Mark. We have one questions from audience in the Zooms. Sure. Oh, I would like to invite, I think one question from Jessica. Okay. Jessica, are you with us? If yes, would you like to, to open your mic and directly ask to the Prof. Mark or Prof. Elsa? Apparently no. Okay, I will read it for you, Mark. So, could the genetic relationship between human and dog parasites be different in other endemic regions?
Yes, absolutely, Jessica, it could. As I just said earlier, it might be quite different in Africa, where the parasites in dogs and people. But so, until something else study there, we won't know. Same in South America as well. People infected with dogs and people infected with Stercoralis there, again, it might be quite different. Studies need to be done, but if you're interested in doing the studies, all I'd say is to make sure we have St. Patrick sampling. So same place for people and dogs, then you can work this out.
Okay, thank you very much. Is there any more questions? No? So actually the genetic markup of this worm is actually affected by many other factors from their sexual reproduction method that maybe they use. Or maybe I was just wondering whether if this any genetic mutation or variants and then these variants is preserved due to because the parthenogenesis will tend to preserve any variants, right? There's no recombination on it. And what happens if, for example, these cross infections from dogs or humans that you present might be caused on that and they can adapt? Maybe would that leakage might be preserved later on and we can obtain worms that can infect both species, dogs and humans, or maybe other hosts?
So yeah, that's a really good question, Yogi, and I didn't really go into that enough. But I did a little bit here. So the parasites seem to be mainly asexual, so it's rather rare sexual reproduction. So we had that parasite, the cross-infection, dog infected with the human parasite. So that means the worms got into the host. There's no sexual reproduction inside. So the So for the chance for sexual reproduction, it has to happen at least these afterwards. So the eggs have to develop into free living adults, two strains, and they have to cross. So there might be quite a lot. So a lot of cross infection might never give the chance for hybridization. So really, so also if you look at the admixture analysis, lots of these parasites have no admixture, and that's probably because sexual reproduction is relatively rare. You did an equivalent thing for a sexual population. Every genotype would have a mixed ancestry, pretty much. So these patterns are really a consequence of the asexual reproduction. So that's probably this asexual reproduction is probably slowing down the well, it's favoring the divergence of the parasites, I think. So if you think about it, if genotype goes across by mutation, it can diverge. This happens only rarely. So if there's a lot of sexual reproduction, these would be mixing all the time. Because there's no sexual reproduction, if they do cross-infect, it's hard for them to keep together in divergence. That was a bit of a muddle answer. I'm sorry about that.
It's okay. Thank you very much. Please give big round of applause for both Prof. Elsa and also Prof. Mark. And yeah, I think before I'm closing, I usually in Indonesia we have with what we call pantun, Mark. If you know, it's like in idiom. What do you call pantun in English? No, poet. No. Yeah, I try to be. I try to make a good one. I'm sorry if it is not good. Okay. Nanti siap-siap bilang cakep ya. Woi, terima kasih. Oh enggak ya. Padahal belum kenal saya. Ya. Cacing stronggy jalan di kaki. Oh.
Cakep.
Cakep no. Kalau ada stronggy ini di kaki kan gimana nanti infeksi. Cacing stronggy jalan di kaki. Oh no. Saya ulang lagi ya. Cacing stronggy jalan di kaki. Thank you, Mark Viney. Sampai jumpa di lain hari. Do you know? Okay. Yeah. Thank you very much, Prof. Mark and also Prof. Elsa for the amazing and wonderful knowledge. Now I will give the sessions back to Master of Ceremony.
Thank you very
This is the degree that required no educations. Master of Ceremony. To get Master of Biomedicine, we have to take the
2 years course.
2 years course, yeah.
With the
True.
research and everything.
But
yeah.
for Master of Ceremony,
Thank you very much,
Dr. Rima, yeah.
the gentleman. Thank you. So we reached to the end of our session for today. Again, we would like to thank you for your patience during our technical arrangements this afternoon. Also, participants on Zoom, thank you very much for joining us. It's very nice to see you online. I hope that we can meet in person one day. Thank you. And I think we have a souvenir for Professor Mark Viney. So, Prof. Elsa Herdiana, would you mind to hand over the souvenirs from Monggo, please, would you mind move to the center? It's not staged, but yeah. Yeah, we are for the photographing. Fotonya boleh dari depan, Mas, sebentar. Sekedap. Please hold on for a moment. A surprise for Professor Mark. Thank you very much for the insightful topic of today's lecture about the Strongyloides stercoralis and everything. Thank you. Thank you very much, Prof. Elsa. Prof. Mark, thank you. And we would like to thank you, all of the study programs and the departments who already collaborate with us at the Department of Parasitology, the Master of Tropical Medicine study programs, and Master of Biochemistry
Medical Science Study Program and the Center for Tropical Medicine. Thank you. We do really hope that the sea worms project. Do you still remember what sea worm stands for? Worm sea, sorry. No? What? Watch out, blah, blah, blah. You should check out. You should check out the website. They have website, right? I think I will announce the, hang on, I have the website. Yeah, www.wormsea.org. Worm, that's, and then sea.org. So you can find out more about the study there. Thank you very much. My name is Rima. I'm signing out. Wassalamualaikum warahmatullahi wabarakatuh. Sampai jumpa.
Mungkin kita foto
Grup foto ya.
grup foto ya, betul.
Picture first before we get back home. I think it would be better if we have it there. Mas Fikri will be from here. The lampu. The light.
Mungkin teman-teman yang belakang bisa merapat kali ya, biar terlihat dalam satu frame semua. Ya. Yang di samping utara dan selatan mungkin di sisi sini, ya. Jadi biar terlihat. Atau kursi belakang, kayaknya seru ya. Kita, oh ya, kita foto di kursi kuning itu ya, itu kayaknya selalu jadi tempat untuk foto ya. Oh, tempat foto. Ya.
Kita
Please,
pindah ke
we take a picture, yes, at the back,
the orange chair.
the yellow chair.
Di tengah nanti ada Prof. Elsa, kemudian Prof. Mark, Mbak Risha, dan Mas Erwan, serta Dr. Alvin dan Dr. Yogi. Bisa di tengah atau menyesuaikan di belakangnya juga boleh. Kameranya aman, Mas? Masih banyak es kopi ya? Masih kosong. Sini kelihatan kan Mas Fikri? Oh. On my count. In 3, 2, 1, cheers! 3, 2, 1, Strongyloides! 3, 2, 1, step on. Thank you all. Take care. Have a good day. Terima kasih banyak Ibu Bapak yang sudah bergabung via Zoom guest lecture hari ini. Cukup sampai di sini. Terima kasih banyak atas kesabarannya yang sudah bersedia menunggu hingga technical arrangement-nya kembali seperti sedia kala. Saya Rima pamit. Terima kasih. Wassalamualaikum warahmatullahi wabarakatuh. Sampai jumpa.